We're at part 9, and it's the immune system, page 784, right? And we were just talking about class 1 major histocompatibility proteins and how this is one way of identifying individuals that might be genetically predisposed to different sensitivities. And so whether it's the flu, COVID-19, and again, it might be the way in which we process pharmaceuticals. So as I was getting into telling you that the research regarding a lot of disease states as well as the usefulness of various pharmaceuticals has been carried out mainly on men and not so much on women. So we don't really know in some cases what the pharmaceuticals will do in the bodies of women compared to men. We already know that heart attacks present quite differently in women than they do in men. Men have a lot more, for instance, peripheral types of pain that could be referred to as a phantom kind of pain that goes down the left axilla, down the left arm meridians that relate to the dermatome T2, whereas women don't seem to have that consequence so much. They have a very different development. They have fatigue, and they have actually direct pain in the chest. So there could be very different presentations. Okay, the T cells actually become, how shall I say, informed by the class 1 major histocompatibility complex, and they develop their, how would I say, immune competence from studying the major histocompatibility 1 complex of the individual. Okay, so we get on to class 2. Class 2, it says here, give us a clear definition. It says class 2 major histocompatibility proteins are typically found only on the surfaces of cells that present antigens to the CD4 cells. Who presents antigens is going to be the dendritic cell, because that's the antigen presenting cell. The macrophage, which is the more overview, how to say, the larger category of what is the smaller category, dendritic cells. It's an antigen presenting cell. The B cell is also going to present a class 2 situation, and so it's going to be requiring to have co-stimulation. Okay, so these are special cells. We don't mess with those cells. These are class 2. They sort of have extra, how would I say, dignity in the system, as it were. Okay, then I'm not going to bother you too much. I think I've also given you enough information that if you need to look through some of these other figures, these are little subsets of what I've already presented to you in the handout that I gave you. I pointed out to you that there needs to be co-stimulation. You need to see the self, and you also need to see the foreign antigen in order for there to be a complete configuration in stimulus that releases interleukin number 1. In the absence of that, we have something called anergy, and so basically the cells tend to stand down, as it were. The T8 cells that would be cell-mediated cytotoxicity would be sort of standing down from that particular item. There's a lot of that going on actually in the digestive system. There's a lot of what they call anergy or tolerance in the digestive system, because the tract is so vast in terms of its surface area. So you need the double handshake in order to get to where you're going. Right? So then we press on. You might want to be reading about what are cytokines and what's a cytokine storm. That's a little bit beyond the scope of what we're doing here. But Nevertheless, cytokines are all the molecules of interest, the interleukins and all kinds of other regulatory molecules. If you get a storm of molecules that respond in a way in which inflammation is not going away, and we get this sort of positive feedback, as it were, mounting stimulation, then the immune system just goes all out, and that can be ultimately destructive, especially to the people who are immune compromised. Alrighty then. Let's press on into other matters here. Here we have the Dr. Merib's workup of what I've already given you. And it's just basically, she sort of spreads out the constellation a little bit more. But basically the idea here is we have the antigen-presenting cell. We have the naive T cells. We have the CD4 T cell, the T helper, which ultimately can get activated initially by interleukin-1. It continues to self-activate with interleukin-2. It sends interleukin-2 over to the T cells that are going to engage in cytotoxicity. So this is just her way of doing the program. We also activate B cells. All right, so she's kind of spread it out across the page a little bit more and differently than I have. So you can sort of compare her diagram with mine and decide which one you like the best. Okay, then we get on to immunodeficiencies. We have a condition that's referred to as severe combined immune or immunodeficiency, SID, or however you want to make that acronym pronunciation. And it's a deficit of B cells and T cells. There are individuals who are incapable of having functional B cells and D cells. They have essentially no immune system. They are bubble children. We've heard about the bubble boy who lived for some period of time. Couldn't live without hugs. I would submit to you that there is a failure to thrive without human touch. And human touch is just one of the most enormously exquisite and essential things for the development of human life, especially early on. And they even have experiments, psychology experiments, by covering over a wire mesh pretend mama with a fur coating, and as long as the baby monkey can touch fur and hug something that sort of seems like a mama, like a teddy bear, then there is less of a failure to thrive, less of a failure and more of a likelihood of thriving. Right, so hug your trees, hug your teddy bears, your G.I. Joes, if especially if Joe's wearing something furry on him. But at any rate, that's the idea, is that you want to have that contact. Okay, there's other types of immunodeficiency, and HIV is one of those. HIV specifically targets the CD4 receptor on T helpers, so imagine how naughty that is, that you have a disease state, and a virus that actually takes out your T4, your T helper cell, and that's naughty. So that's what's going on with HIV, and what leads to some of the later stages of the disease. Okay, so it's targeting the CD4 or the T helper cell, something to bear in mind. Okay, I'm not going to get into all the epidemiology of AIDS. It's an area that I was extremely interested in the 1980s and 90s. We now have medications which hopefully could also be applied to other viruses of the RNA persuasion. HIV is an RNA virus. COVID-19 is an RNA virus. And it would seem to many that David Ho's protease inhibitors might be a method of treatment. So one of those things that you want to keep in mind. Autoimmune diseases are plentiful, and ultimately rheumatoid arthritis is one such, going after the joints of the body. Myasthenia gravis is one in which the receptor for acetylcholine for muscle contraction is targeted. multiple sclerosis, which targets myelin sheaths in the central nervous system. Now there's also a peripheral version of MS that's been known to target the myelin sheath in the periphery. Graves' disease, which involves the thyroid gland and targets oversecretion of thyroxine. And then there is type 1 diabetes, which targets the beta cells of the pancreatic islets, the islet of Langerhans, and knocks out the ability of the pancreas to produce insulin or adequate amounts, sometimes very little or virtually none. Then we have systemic lupus erythritis, erythematosus, okay, SLE for the easy way out. And basically, this type of targeting can go to virtually any tissue, even DNA. It can be targeted, so it's quite the horrific disease. Glomerulonephritis is one that we're going to be dealing with in the particularly in the immunosensitivity realms. Treatment is quite tricky and involved, and I won't even begin to address it so much except to say that we can relieve certain deficiencies by adding things in. And